So this is the architecture of this data sheet is lifted from the TTL databook from Texas Instruments. If you're familiar with that, you got the transfer function, the response time, the specificity of the receiver to different input signals. The one thing that's different is the bottom right plot, which is the evolutionary stability of this device. How quickly will it mutate as you go from one generation to the next? Yeah. What's the license and so on. So one of my projects for now is starting a factory that makes lots of parts and puts them in the public domain. So it's a lot of work to do that. And so to sort of transition to the last part of the talk, as we've been implementing these very naive and primitive lessons from other forms of engineering in support of genetic engineering, what we're finding is people like to make stuff. So here are students going back to 2003, 2004, 2005, 2006. These are students who participate in something called iGEM. iGEM is the International Genetically Engineered Machines Competition, which is a worldwide genetic engineering competition for undergraduates, in which teams of students from around the world compete over the summer to design and build a genetic engineered machine of their choosing, so long as it's a constructive thing. And so here's the world map of the teams from 2006. And you'll see we didn't have a lot of teams from China, so then we went out to China and we got 5 schools from China to come in. And so here are the students from this year. And so, for example, the team from Melbourne, Australia showed up with some new BioBrick parts. They wanted to change how bacteria float, give you that option. If you're working on some project, I don't know what, but you want bacteria to float or sink or remain neutral, well, here's the part for you. This piece of DNA encodes a little protein balloon that is somehow gas impermeable, and the protein balloon boots up inside cells.